Okay, so we're starting with Cathy McGlynn. Cathy McGlynn is the Aquatic Invasive Species Coordinator for the New York State DEC. Her job is to help implement the New York State Aquatic Invasive Species Management Plan and lead the New York State DEC Aquatic Invasive Species team. Team oversees the statewide watercraft inspection steward program, executes control and management projects, performs research, conducts monitoring and surveying, and provides education and outreach to multiple target audiences. Cathy received her PhD from the Department of Ecology and Evolution at the State University of New York at Stony Brook. And now I'm going to give the mic over to Cathy with her presentation on the Peconic River Invasive Aquatic Plant Control Project. Take it away, Cathy.
Thanks, Melody. So good morning, everybody. I'm happy to give some updates on this project. Thanks for the opportunity to share this information. So today I'll be sharing with you a bit of background information as well as most current information about the Peconic River Invasive Aquatic Plant Control Project. So again, this is just the overall outline of what the talk will include. And we'll jump right in. So the Aquatic Plant Control Project originally started out as a focused project on management of Ludwigia peploides, which has many common names, so I'll just refer to it as Ludwigia throughout this presentation. It's an invasive ornamental, and it is currently outside of its native range when in New York, but its native range is fairly close by. It is a federally listed noxious weed, and it is prohibited by New York's Part 575. It's an aquatic wetland plant. It's herbaceous perennial, has creeping stems, 5-petaled yellow flowers, which are in bloom all summer long, reproduces mainly through fragmentation, and also produces seeds, the viability of which we're still wondering about. And then, as part of our last year's management effort, we included an additional species to the project, which is Hydrocharis morsus reynae, or European frogbit. So this is an ornamental as well, native to Europe and Northern Asia. It's a free-floating aquatic perennial plant, reproduces through turions, which are overwintering buds that grow in leaf axils, and through plant transport, and it is also listed in Part 575 as prohibited. So overall impacts of invasive aquatic plants, which I'm sure folks have already heard quite a bit about, include they form dense mats, which can displace native aquatic plant species. When those mats decompose, the bacteria that helps to decompose them can reduce dissolved oxygen in the water and also impact pH. There could also be a reduction in water quality for wildlife, as well as this dense vegetation blocking waterways and increasing sedimentation. And those mats can also interfere with flood control, drainage systems, impacting navigation and recreation and real estate values. So there's a lot of impacts to these species. So the focus area for our project is Peconic River, which is 15 miles long. I believe is the longest river in Long Island, sorry. So it extends from the bogs and wetlands of Brookhaven National Labs out to the dam east of Grongabel Island in Riverhead, where it becomes an estuary. And it does flow through a good portion of the Pine Barrens. So the need for management has been growing. So Ludwigia was first reported in the Peconic River around 2003, and efforts to hand remove began in 2006. They've averaged about 300 hours a season to remove anywhere from 4 to 60 cubic yards of Ludwigia. And in July of 2020, I know that DEC and volunteers removed about 1,800 pounds from the river. And just demonstrating that the many years of hand pulling have been unsuccessful in preventing the spread of this species. So it was first discovered, again, in 2003. It was confined to Suffolk County. Currently, the infestation spans from Connecticut Ave to Peconic Ave. There are some additional sites nearby, a private pond at Windcrest East, as well as Artist Lake. And there is one other known infestation that was reported on Staten Island. So in September of 2020, well, actually, I'm going to go back to summer of 2020, we reached out to stakeholders to see if we proposed a pilot project, how folks would feel about us doing some control of Ludwigia on the Peconic. And we got some positive responses, and we're keeping people apprised of our plans. And then we met with the Suffolk County CAC in September of 2020. and gave them a presentation about our plans to do a pilot treatment using 2 herbicides in an area under 1 acre, which means we wouldn't need a permit. Or so actually we would need a permit because it's in an area that's larger than 1 acre, but the treatment area itself is small and is solely owned by DEC. Sorry about that. So solely owned by DEC, so that would make things a lot easier for us. It was at the Edwards Ave fishing access. And we found that the treatment was effective, as noted in the photo on the right. So we had mentioned to the CAC that if the treatment were effective, we would plan to expand the treatment across the entire freshwater portion of the Peconic River. We had notified them of this in September of 2020. So once we had our pilot study efforts completed and our results for 2021, we did move forward with plans to expand the treatment to the entire freshwater portion of the Peconic. And here we divided it up into 9 different sections for treatments and surveying purposes. In 2022, again, our first year of the expanded control effort, we targeted only floating water primrose with a mix of 2 herbicides, Mazamox and fluoroperoxifene benzyl. The treatment was effective with a few points remaining post-treatment. In 2023, we conducted our pre-treatment surveys in the week of June 26th and the post-treatment surveys the week of August 21st. Treatment itself was conducted during one day, July 11th. So just want to back up a little bit and emphasize that having aquatic plant surveys for both post and pre-treatment conditions is very important to us for these projects. The very first post-treatment survey serves as our baseline for comparison to the last post-treatment survey of the project, which helps us to gauge how effective our management methods were. In addition, each year when we conduct our pre-treatment surveys, the data from those surveys dictate where we do the herbicide treatment, which helps us to keep our herbicide use to a minimum. And it will allow us to gauge how effective our herbicide treatment was for that year on the target plants as we do our post-treatment surveys, and also allow us to monitor for non-target plant impacts in the post-treatment surveys. This is just an illustration of the surveys that are done, the combination of point intercept method with rake toss surveys. So basically, we'll set up a layer in GIS. They'll place a grid, either 50 or 25 meters by 25 meters or 50 by 50 meters. That grid will be placed over an area. Where those grid lines intersect are the points where the rake tosses are conducted. And you'll see these points here, just again, the pre-treatment survey, the Peconic, and you'll note that we have a grid across the entire section 2 of the Peconic. And our goal here is to get the yellow in all of the points, which is 0 presence of Ludwigia. But moving back to our categorization of what you find on the rake toss, you toss out your rakes twice. You will estimate the density on the rake. So trace would be a fingerful or 2, sparse, maybe a handful. I know sparse is a little high in this illustration, but it should be less than that. A medium and dense abundance overall, and then we'll estimate by species and again ID what species are present on the rake. This can also be accompanied by visual surveys in some cases. In other projects, we do snorkeling, scuba diving, in addition to using cameras and biobase, which will help target high density vegetation areas. In 2023, after a successful season treating Ludwigia in 2022, we added the European frogbit to our hit list, pun intended. And this because it was often associated with Ludwigia and can be very challenging to hand pull throughout the river. So as was mentioned in a previous talk, this species was not on either herbicide's list, so we obtained a FIFRA 2EE recommendation for its use on using both of these herbicides. So what we needed to do was we actually reached out to the herbicide manufacturer, CPRO, as well as a non-biased lab at University of Hartford to conduct lab tests on sensitivity to both herbicides using European frogbits, which we collected and provided to both labs. And that data was given to DEC to review so they could issue a FIFRA 2EE recommendation. So we found that leaf damage over time indicated that we could use a combination of both Mazamox or brand name Clearcast and fluoroperoxifene benzyl, which is Procellacor EC brand name, 32 fluid ounces each, seemed to work quite well. So that was what we presented for our FIF for 2EE. And as was discussed earlier, our contractors wrote up the Article 15 permits for this project. In February, that Article 15 notification letter was sent out listing a tentative treatment date of July 24th, and we had additional notification letters sent out June 28th with the new treatment date based on weather and scheduling. And the day of treatment, as I had mentioned, was one day only, July 11th, sorry. So products used, a little more detail about the tank mix, fluoroperoxyfenbenzyl and Mazamox, along with a non-ionic surfactant and an indicator dye. So the non-ionic surfactant will help the herbicide stick to the leaves because it was a foliar application using spray. So most of the herbicide is taken up by the plants, particularly on sunny days like we had on July 11th, but some percentage does make it to the water surface, which is what we monitor using water samples. So a little bit more about the herbicides that we chose. Both are rated by US EPA as reduced risk aquatic herbicides. There were no restrictions on swimming, eating fish from treated water bodies, using water for drinking water, or irrigation of turf during that time. These herbicides were selected because they are effective control of target species. They have low use rate orders of magnitude less than more comparatively older herbicides. They are absorbed quickly by the plants. They have a low aerial drift potential, and the day that they were applied in 2022, there was very little wind activity. They're highly selective for those target species, and no significant adverse impacts on non-target plants were anticipated because we were using levels that are well below the concentrations that were listed for some impacts by EPA. So again, the application was dictated by the points and polygons for our pre-treatment aquatic plant survey maps, and it was a foliar application using a liquid formulation sprayed onto the leaf surface. Water sample collection began 24 hours after treatment, and those samples were analyzed by a CPRO lab for the presence of the two herbicides at 21 sample points. The detection limit for the test they used was 1 part per billion. So looking at the two herbicides, we see that we had no non-detects for both herbicides 1 week after treatment. So Procellocori C was a non-detect the day after treatment, and Clearcast or Mazamox was a non-detect fully on July 17th, but was a non-detect at 18 of the 21 sites on July 12th. In addition to this, we do macroinvertebrate surveys because there is concern about impacts on non-plant life in treatment areas, and macroinvertebrates are known to be quite sensitive to herbicides. So we did macroinvertebrate surveys 1 week prior to treatment at these 3 locations, and then 48 hours after treatment and 7 weeks after treatment. So looking at the site locations, moving from east to west, we have the Lower Peconic Fishing Access Site, or Site 1, the Calverton Angler Access Site, the Edwards Ave Angler Access Site, Site 2, and Site 3 was the Calverton Angler Site. So we conducted kicknet surveys and filtering, and these specimens were then placed in jars to be analyzed in the lab, where they were identified into families and orders. Any data that included families with at least five observations were used in the statistical tests, used a chi-square test to determine if sites were significantly different in composition, pre and post. So here are the orders and families that we looked at. And here are some of the data. So I'm going to unpack this graph a little bit. So the top row is the pre-treatment survey. The middle row is 48 hours after treatment. The lowest row is the 7 weeks after treatment. And then the individual columns are the 3 sites where we sampled. So site 1, as you'll note, had an increase. Well, actually, it had as all sites here, they had a decrease from pre-treatment to 48 hours post-treatment. We noted qualitatively a decrease, but then an increase to abundances above the pre-treatment levels 7 weeks after treatment. So we've noted that for all the sites. So looking at individual sites, So site 1 in Lower Peconic, we had a dominance of Trichoptera or caddisflies, larvae and amphipods, and additionally annelids such as leeches and worm-like organisms were notably abundant. And looking at 2021, which was before any treatment, was dominated by amphipods and caddisflies. And then post-treatment in 21, dominated by amphipods and mayflies. And remember, the post-treatment, the treatment itself in 2021 was relegated to under 1 acre of the river. Site 2, the Peconic Lake Dam site, in 2023 displayed a more evenly distributed community with lower overall abundance. Diptera were a little more abundant than other orders, but not in a statistically significant way. In 21, this site was dominated by amphipods, and post-treatment also dominated by amphipods and mayflies. And the Calverton Angler Access site, in 23, we had isopods marginally dominating the community, but this site exhibited the lowest diversity among all 3 sites. And in 2021, before treatment, there was limited diversity with amphipods present, and post-treatment amphipods and mayflies were present. So our overall conclusions, the chi-square test did not find a statistically significant difference in community composition among the 3 different sites. Therefore, no significant treatment effect was found overall. Any differences in abundance that we saw qualitatively in the graph that I described earlier could have been within a margin of error for collection or due to other factors, and we'll do more research this coming season. Another issue that was brought up by the public and has been a concern for multiple herbicide treatment projects that we've done is the concern of dissolved oxygen reduction. So what happens is the plants will decompose, and in some cases folks are concerned they'll decompose rapidly all at once, and they'll reduce DO dramatically and impact fish and other wildlife. So we'll note that herbicides are used before the maximum growth of biomass has accumulated, usually earlier in the season, to mitigate the dissolved oxygen reduction. And I also want to note, just because it's important, that if there were no treatment, there would still be an enormous amount of biomass at these sites, and they would decompose at the end of the season, reducing DO. So our DO monitoring site this past year was 1 location. We located it in a patch of Ludwigia pre-treatment that was about 100 feet from the launching site in the Peconic River Dam. Loggers were placed, 1 within the patch, 1 at the edge, and 1 outside of the patch at a distance of 2.5 meters from each other. So noting the overall trend in the water temps from treatment day until I think it was early November when the loggers were removed from the river, there was a downward trend in temperature, which likely means there's more of an upward trend in DO. So our DO analysis, we used a paired t-test, which found statistically significant differences in dissolved oxygen at all 3 locations within the patch. So unpacking this a little bit, we have the green line representing the innermost logger, the red line representing the logger at the edge of the patch, and the blue line representing the outside of the patch logger. So you'll note that the dissolved oxygen is increasing as the months go by and the temperatures drop outside of the patch. And the innermost or green line, the dissolved oxygen is dropping quite rapidly after the initial the herbicide treatment, and then it will hover around 0 from about mid-July until November. And then most interestingly to me is the logger on the edge seems to have quite sporadic increases in DO and then drop-offs until around October, where it then starts to hover around 0, except for that 1 little blip at the end of October. So what we're seeing, we think, happening here is the vegetation is decomposing and reducing oxygen within the patch. And there was also the arrival of algae in that area where the patch was. So that too decomposed toward the end of the season, further contributing to the reduction in DO. So we will, again, do additional work on this to study the DO a bit more. But I do also want to share the results of our surveys. So we did our pre and post-treatment surveys this past year. And for Ludwigia in pre-treatment, we had 17 of those rake toss points had some degree of Ludwigia. But post-treatment, there were only 4 points found. For European frogbit, pre-treatment grid, we had 48 points with presence of European frogbit. Post-treatment, we had 6 points. This is a little bit of a Ludwigia patch that was found post-treatment, just noting that there were some areas that still need some additional help this coming season. I keep saying next year, this coming season. And this is also a demonstration of our grid where we had pre-treatment survey. You're noting 3 points that are blue, which means they have trace to sparse amounts of Ludwigia pre-treatment. And we found 4 locations in that area in September. So we are, again, going back to those locations and redoing the survey in June before treatment starts. For European frogbit, we have the pre-treatment grid. We're seeing a lot of blues here, even some medium densities. And then post-treatment, we're seeing 5 points, which are light blue, meaning they're trace densities of European frogbit. Or sorry, Ludwigia. Oh, no, it says European frogbit, sorry. Yep, okay. So overall, the 2023 outcomes are very positive. We had a decrease in herbicide use and definitely a decrease in coverage by both invasive plants. So in 2022, we used imazamox, about 15 gallons of imazamox on 61 acres of infestations, and 14 or so gallons of clearcast on 57 acres. In 2023, we used 6 gallons on 25 acres for Procellacor and 6.25 gallons on about 25 acres for clearcast. So this is a really positive outcome for us. Plans for 2024 include adding an earlier macroinvertebrate survey in May because we wanted to have another baseline for the families and orders that are present at each location, and doing additional DO monitoring at 2 other infestation sites. And we're also talking about putting in loggers at a native plant patch as well, just to compare the amount of DO changes that occur naturally in native plant habitats as well during the end of growing season. In addition, since we seem to have good efficacy for water primrose and European frogbit, we'll be wrapping in parrot's feather now, which is also covered in the labels of both of those herbicides that we're using, imazamox and fluoroperoxib benzyl. So that is an EDR species on the Peconic. So we have 10 points found in the post-treatment survey, which we'll be treating this coming season. More plans for 2024 include letting people know that 2024 is our last contracted herbicide treatment season. So we'll be doing our best to make sure we hit all the points that are still having some lingering densities. And then from 2025 through 2026, we still have anaerotic research and our own DEC staff monitoring Ludwigia, frogbit, and parrot feather, as well as other invasive and native aquatic plants. And again, as I always emphasize, our project method is focusing on adaptive management. So the data from previous seasons informs the next season's plans. Same season pre-treatment data will determine final treatment locations for that season. And of course, we're constantly prepared to adjust depending on what the data indicate. So our plans for this year, we already have our Article 15 permit done. It's been submitted. I'm sure the notification letters were out fairly soon. Our contractor's working on that. In May, we'll install the dissolved oxygen loggers and also conduct the first macroinvertebrate survey. In June, there'll be a pre-treatment aquatic plant survey, pre-treatment and macroinvertebrate survey again, sending out additional notification letters with that finalized treatment date. In July, we'll have the 1 day of treatment, then water sample collection and analysis, and then the mid-season or the 48-hour macroinvertebrate survey. And then August will be a post-treatment aquatic plant survey and potentially a post-treatment macroinvertebrate survey as well, the 7 weeks after treatment. In September, October, even November, we'll do the retrieval of the DO loggers. We'll compile our data and analyze it. And then in December, we'll have our stakeholder meeting. So I probably whipped through this very quickly, but I'm happy to answer more questions. And I have additional slides for frequently asked questions if folks have those questions. So Heidi O'Riordan is our regional contact for this project. If you have questions that you think of later, feel free to reach out to us and ask away. Happy to provide more information. And thank you again for the opportunity to talk about this project.
Thank you, Cathy. You took on a challenging project, but it sounds like it's going very well. looks good. Looks great, really.
So far, so good. Keeping fingers crossed for this season. Yeah,
Have a question if I may start.
sure.
After 2024, after herbicide, are there plans to manually remove any small infestations that might remain or pop back up or something?
That is likely if something becomes a little larger. Most of our staff, not our Region 1 person, but most of the other staff we have on board now will be certified applicators in Category 5A. So we would be able to help out with that too.
Okay, all right.
And
So you
we
could still
started,
herbicide afterwards if anything comes back.
yeah,
Right.
we of
Yeah.
course we'd have to fill out our own permits, but we are likely, and as of next year, most of us will be certified in Category 5A, and DEC now has a commercial permit for herbicide purchase and distribution. So we essentially just buy our own herbicides and then
Oh,
divvy
good.
them up through our applicators.
All right, cool. Thanks, Kathy.
Sure.
There's a question in the chat.
Okay.
Was the 2003 control work that TNC had done in the Peconic River, is that
I'm not sure what that
included in
is.
any of the data or whatever? Was the 2003 control work,
Oh,
the original work that TNC did?
I actually didn't know there was TNC use of herbicides
All
in
right.
Peconic. Okay, so that's news to me. Please share.
All right. Well, we might have a little bit on that, but we can
Okay.
find out more.
Okay.
I think they did a pilot somewhere up on the north side of the river somewhere.
Was it on Ludwigi as well?
I believe so.
Oh, okay. Interesting.
All right. Well, that was a question in the chat. Any other questions do folks have? You can put it in the chat. There's not many people here. You can just unmute too if you want. I mean, there's people, but it wouldn't get too out of control if you just unmute and ask a question.
Yeah, this is Steve Young. That was my question. Because I remember seeing a report from TNC when they did herbicide use. I thought it was in the 90s, but maybe it was 2003. And there's a report on it, detailed report, and which might be in your files under that species.
Yeah, I think we have something, Steve. We'll dig it out and share it with Kathy.
That'd be
It might be
great.
useful for something.
Yeah. So I do want to add that we are starting to think about restoration and what that could look like here. So maybe that was sort of a conversation I started when we had the LISMA meeting a few weeks ago. It's like I really would like to have a brainstorming session about what that could be in Long Island.
All right, sounds good.
Okay.
Any other questions or comments? All right, well, thank you very much, Kathy. That was
Thank you.
very interesting. It's good for all of us to be on top of that, how that's going. It's a great project. And I can add that LISMA is doing monitoring in the Carmen's River. Directly ourselves, we've not found Ludwigia peploides in the Carmens. And we do check IMAP and iNaturalist for the Nisquag River as well. So we're keeping an eye on it so it doesn't pop up anywhere else. All right.